Good morning. Could you indicate that whoever is watching can actually see me online? Good morning. This is General Surgery in Quarantine, and today's lecture or seminar is on molecular biology for surgeons. This is the second of my two-part series, and I'm going to wrap it up with today's lecture. Before I start, I'd like to allude a little bit to the COVID pandemic and its similarities in some ways to the complexity that exists within a cell. From my first lecture on molecular biology for surgeons, you would have realized that there are so many interactions, so many different assumptions that you have to make when you think about a cell, that it's difficult or impossible to predict in exactly which way a particular intervention might go. So you would have realized that several of the models which epidemiologists have put forth in various forums on the internet to government officials and so on, those models are now being shown to be, in some cases, grossly off target, in some cases wrong, in some cases, in fact, so wrong that people are thinking, what were these epidemiologists thinking about things like mortality rates and so on, including the WHO. So many of those models we have realized are wrong or off target. That's not surprising, and it shouldn't surprise you because biological systems like those within a cell are extremely complex. And the complexity is part of the wonderful science that is behind this. But we don't understand all the complexity, and certainly we don't understand the way in which the complexity can in some way be broken in a way that makes for things like flattening the curve or abolishing the curve and so on. So it's a complex system, and molecular biology is a complex subject. Now, you would have heard of DNA a lot when you were reading or studying about molecular biology. But as I told you in the last lecture, molecular biology is not just DNA, it is also RNA, it's also the proteins that DNA and RNA make, and it's also the molecular interactions between DNA, RNA, protein, and these various systems interacting with each other. So what does RNA do? RNA, as you know, is a protein synthesis molecule. You have three types of RNA, and this is well known to you: messenger RNA, transfer RNA, and ribosomal or rRNA, M, T, and R RNA. Now, RNA has become as important as DNA in molecular biology in the last 20 years. We know that they do a whole lot of things other than code for protein. There's a whole area of science that is devoted to non-coding RNAs. In other words, RNAs that don't make protein, but which have a regulatory function in gene expression. Excuse me. Now, you would recall that a nucleotide is a nitrogen base, and then there's a backbone to the base of a sugar and a phosphate. So 3 nucleotides constitute a codon, which codes for 1 amino acid. Now, the sugar in DNA is deoxyribose, but in RNA, it's ribose. That's the first way that RNA differs from DNA. The next way in which it differs is that the bases in RNA are different from those in DNA. In DNA, you have A, G, C, and T, thymine, but in RNA, you have A, G, C, and U, uracil. And finally, RNA is not double-stranded like DNA. It's single-stranded, but the strands can double up and then double up again into various 2-dimensional shapes. How does RNA make protein? It's a 2-step process. First, there is what is called transcription and then translation. Transcription is something like, I was alluding to this in my last lecture, how a scribe takes down you have these old stories of scribes writing down instructions from, let's say, the king, and then taking it out and making those instructions known. So the writing down of instructions is transcription. Making it known and translating it into a protein is called translation. So during transcription, the enzyme RNA polymerase initiates the process by which the instructions from DNA is read. And as the mRNA moves up and down the DNA strand, transcription is terminated by what is called stop codons. So these are what they are referred to as the cap and tail system. So once that protein instruction is read from DNA to RNA, the cap and tail codons make it a stable molecule so that it is not degraded when it passes out into the cytoplasm. Translation is the process by which, as I described to you, the instructions on the mRNA is translated into a protein through the help of tRNA. Translation occurs in the cytoplasm on the surface of ribosomes. Now, ribosomes also have their own RNA, their cytoplasmic structures, but on the ribosome is what is called rRNA. rRNA is like a catalyst. It acts with tRNA to do the work of translation in the making of a protein. Now, how does translation occur? The mRNA attaches, the mRNA attaches to tRNA and then reads the amino acid or codon instructions to tRNA. So tRNA then brings the amino acids together, and each amino acid is linked to its neighbor by a peptide bond. After one codon's information is read and the corresponding amino acid's peptide bond is formed with its neighbor, the tRNA detaches, leaving an amino acid behind. And each time the tRNA acid binds, an amino acid is added to the growing chain of amino acids by peptide bonds. Now, the process of translation then stops when tRNA and RNA encounter, you remember I talked to you about those stop codons, the cap and tail system. So when they encounter the stop codons, the process of translation stops. A polypeptide chain is released. It is folded. Sugar or other molecules are added, and voila, there is a protein. So on the left of this image, You have what is going on in the nucleus, where the instructions for the protein are read into the mRNA, and then the mRNA goes into the cytoplasm, teams up, as it were, with rRNA, brings the amino acids together, links them with peptide bonds, moves away when the job is complete. Sometimes organic or inorganic substances are added to the peptide chain, and a protein results. Now, this is something that has come to light again in the last 20 years or so, but especially in the last 10 years. So as I explained to you, RNA does not just make protein. There's a whole lot of RNAs for which only regulatory functions have been found. And there are lots of RNAs actually for which we don't know the function yet. But there is a RNA that is called RNA interference, or RNAi, which is a regulatory RNA that binds to mRNA. So there's mRNA, it comes with instructions, and then the iRNA or RNAi binds to it and stops it from making it a protein. It was discovered in 1998, and there are several types of RNAi. There's a miRNA, or the microRNA, which has been associated with cancers, heart disease, immunological disorders. And then there's the siRNA, or the small interfering RNAs, which is another type of RNAi. Now, what is the practical application of this? In fact, there is a lot of research going on mainly in the laboratories of multinational drug companies. Now, that's another big discussion as to why much of this commercialization actually happens in drug companies rather than our universities, although our universities also get billions of dollars in grants. But this is a drug that was that first won FDA approval several years ago. It uses RNAi, and patisiran is a very good treatment for a congenital fatal type of polyneuropathy. It's a very rare polyneuropathy, but it's been treated with a gene silencing drug which uses RNAi. What do you know? Should you know about short interfering RNAs, siRNAs? There's a lot of research going on to the function of genes, the synthetic RNA molecules, siRNA, that treat liver cancer, liver disease, cancers, and viral infections. But I want you now to backtrack a little bit and look at some of the problems in delivering these molecules, which we know can do several great things in a cell, to the cell. So in the laboratory, we know that certain things have certain functions. We know that if that function can be translated, can be injected, in some way made to go to the target organ in the human body, you have a drug. What are the problems there? In fact, there are several problems, and they all fall under three different categories. One is the drug or the delivery system for the molecule has to evade the immunological systems in the body. Secondly, there are enzymatic systems in the body that are a product of evolution and are designed to destroy these foreign intruders. And thirdly, you might evade the immunological system, you might evade the enzymatic degradation systems, and you might get the drug with the siRNA or the RNA or the protein onto the cell, but how do you make it enter the cell? How do you make it latch on first to the cell, and then how do you make it enter it? So these are the three different areas which research is focused on. We know a hell of a lot now about molecular biology, what genes do, what siRNAs do, what RNA does, what proteins do, but how do we get that into a cell? That is a big challenge. One solution is to inject the siRNA molecules directly into the tissue. That's actually being done in age-related macular degeneration. It's actually being done in our ophthalmology department too. It's a drug that the FDA has approved. I believe it's not yet approved for use here, but it can be used in an experimental fashion. I see no reason why it shouldn't be, because there's very good data that actually it works. And patients here have to pay for it, I believe, but it can be done. There are other regulatory RNAs. There's an long non-coding RNA. We don't know what it actually does, but there are more than 10,000 of these. And then there are catalytic RNAs, or ribozymes, which are RNA molecules that catalyze like our RNA does. It catalyzes the work of tRNA in making protein. And ribozymes can be used to block or knock out the function of a gene and thereby prevent its expression. These are, again, terms in molecular biology that you'll hear quite often. And essentially, these are terms you will understand at least partially from my last lecture. PCR reactions, or polymerase chain reactions, are those reactions in DNA which are designed to greatly amplify the quantity of DNA available for analysis. So from a very tiny, infinitesimal quantity of DNA, you can then, by PCR, amplify it to a much bigger amount that will make analysis easier and possible. Now, you know also that reverse transcription PCR is very commonly used. That's using RNA, and then that can be quantified, QRT-PCR. A microarray is nothing but a spread or an array of DNA on a microchip that is used to identify DNA produced by RT-PCR techniques. So essentially, two samples are used, disease, normal. RT-PCR generates what is called complementary DNA for each sample. One tissue's DNA is marked with one type of marker, a red fluorescent marker, the other with a green fluorescent marker. The samples are then combined and then added to a DNA microchip. And then the microchip array then separates it out, and by the pattern of fluorescence, you can make out the relative concentration of the different DNAs. But the commonest way now of studying RNA is by RNA sequencing, which is like DNA sequencing. There's a next generation RNA sequencing technology available. You can actually identify a whole array of RNAs in a fairly short period of time. That, together with all the DNA information available and all the molecular information available, is actually now online. You can see it in what is called the ENCODE project. There's also a cancer gene atlas that is maintained by the cancer institutes in the USA, but anybody can actually contribute to that. It's an international collaboration. This is what the websites look like: the ENCODE project on the left, the Cancer Genome Atlas program on the right. Now, that's the second and equally important part of molecular biology that I was telling you about. And the third is the proteins. So molecular biology, as I was telling you in my last lecture, was initially just DNA. And nobody thought molecular biology was anything other than DNA. We now know that molecular biology encompasses DNA, RNA, proteins, and the molecular systems that interact between them. Whenever you hear molecular biology terms or processes, try and fit it into one of these categories. They will fit. Now, There might be a fifth thing that somebody will discover in the next decade or next 100 years, but as of now, these four areas encompass essentially the whole of molecular biology, equally important. Now, you know that there are about 20,000 protein coding genes in the human genome. But the genome can, by splicing, multiple translation stops and starts, and then post-translation modifications, produce exponentially more proteins. What is a proteome? A proteome is the sum of the more than 1 million proteins that comprise the human body. Now, it's not the size of the genome, but the size of the proteome that determines the complexity of the organism. There are some organisms that actually have more genes, but it is the size of the proteome or the sum total of the proteins that determines the complexity of the organism. Now, the amino acids are 20 in number. You know that. You know that 9 of these are essential. In other words, they have to be supplied by the diet, or in TPN by the intravenous fluid. These are called the proteogenic amino acids. Some amino acids do not make protein. What happens is they make something else. So the amino acids that don't go up to make protein, or the non-proteogenic amino acids like gamma aminobutyric acid, are the non-proteogenic amino acids. If the chain of amino acids is up to 70, they're called peptides or polypeptides. When the chain has more than 70 amino acids, they're called proteins. You probably know that already. Now, another fascinating area of molecular biology, proteins are macromolecules that exist in highly specialized and specific 3D structures. So every protein has its own 3D configuration. It's the folding that makes the protein unique, apart from its sequence of amino acids. This folding is facilitated by hydrogen bonding and the amino acid side group, and there are also things called molecular chaperones that facilitate the folding. Now, proteins often have organic or inorganic cofactors attached. An example would be the iron or the heme group in hemoglobin. The globin is a protein. And organic cofactors related to vitamin B6 and B12. Now, a finely controlled mechanism exists that when a protein becomes tired out or has ceased to function or is exhausted, they basically are destroyed. They're programmed for death. And failure of this mechanism can result in abnormal accumulation in cells, like in Alzheimer's disease. Prions are, so if a protein is normally folded in a particular way, if it's folded in a different way, that's called a prion. And a prion is unique characteristic is this abnormal folding can be propagated in normal protein by a chain reaction. And that causes holes in tissues, like in spongiform encephalopathy. On the left top of this slide is the actual image as revealed by X-ray crystallography of a protein. This is not fantasy. This is not imagined by the artist. This is actually the X-ray crystallography image of a protein. They're not random, and they're specific. Now, what is the practical use of this? This wonderful folding of amino acids that constitutes the protein also means that amino acids can be hidden in areas that people don't know where they are. If you know where they are and what they do, you can design a drug to go and inactivate that area, which is what tenofovir did for hepatitis C. So key amino acids in the active site of the virus producing serine. So the RNA virus produces an enzyme called serine protease. Serine protease is essential for viral replication, but it also inactivates the antibodies in a quite an ingenious way. So the RNA produces serine protease. Serine protease is essential for RNA replication. So by X-ray crystallography, researchers were able to find that there are a couple of amino acids, key amino acids, more than a couple, in a hidden area of the molecule, of the peptide chain, the protein, serine protease. And then they were able to design small peptides which were able to fit into these active sites of the viral protease and inhibit viral replication and therefore cure hepatitis C. When the drug first came out and it was approved by FDA, the cost of one treatment was about $200,000. A lot of money, but if you think about it, it's not just that the drug companies sink a lot of money into this. Some acknowledgement, in my view, should be given to the brilliant research that makes this possible. Should it be $200,000? No. But can it be like $2? Probably not. There has to be some sort of government subsidy, as in Canada, and there has to be a faster way in which it can be approved. There are newer and cheaper drugs based on the same principle as telaprevir that is now available for hepatitis C. Proteomics is the study of proteins. Mass spectrometry is the way proteins are studied, one way in which proteins are studied. It's a little bit like MRI. If you put a magnetic field to a protein mixture, the heavier proteins are deflected less than the lighter ones. So the lighter ones go further, the heavier ones stay closer. Then a protein reader can then identify the different proteins in a mixture. Mass spectrometry is actually very big in research now. Some of you may know that in our medical school building, we have a whole floor, a part of one floor, a big area devoted to mass spectrometry. About 4 or 5 years ago, we recruited a fellow from Greece. I'm not able to pronounce his surname now, but his Christian name is George. George is a mass spectrometry expert, and he essentially has his own lab there. Proteins can be identified by Western blot, which is a similar thing to Southern blot. In other words, gel electrophoresis separates the proteins. It's a kind of a messy separation, but then it's concentrated and made nicely analyzable by blotting it onto a special membrane, and then specific IgG antibodies are used to identify the protein. And then you have immunohistochemistry, which surgeons meet time to time in pathology reports. So proteins in a histological sample can be identified by antibodies. And how do you make the antibody visible to the pathologist? You tag it with a fluorescent dye. And then you look at it under the microscope, and the fluorescent tagged antibodies will show us a different color. As in the HER2 surface receptor protein, which is familiar to you guys. It is essentially a protein that marks prognosis in breast cancer. It is identified by immunohistochemistry. On the top left is a HER2 negative slide, and as you progress, you go from HER2 negative to HER2 3+ score, which is the slide on the right lower corner. immunohistochemistry for identification of proteins. The fourth and final pillar of molecular biology. The first pillar is DNA, the second is RNA, the third is proteins, and the fourth is the molecular interactions and control systems. Each of these are important. Take one pillar out and the whole edifice collapses. The genomic blueprint of all cells in the body is the same, except of course the adaptive cells, T and B cells. So from this genomic blueprint, there has to be specialization, as you can imagine, into various tissues and organs with their own function. And this process has to take place continuously. This requires regulation and a very fine control of what is called gene expression. Gene expression is controlled by molecular interactions and control systems between DNA, RNA, and protein. You will recall that chromatin are the histone proteins plus the DNA, and they look like beads on a string, and these are then compressed and compacted into chromosomes. In order to expose relevant DNA to protein synthesis, there has to be an unfolding of this chromatin compaction. But if the chromatin continues to be compacted, it leads to what is called gene silencing. So in order to make DNA do its job, the chromatin has to unwind and has to expose relevant areas of DNA for action by RNA transcription factors and the initiation of transcription. But if you don't allow it to open, then you can stop the protein from being formed, gene silencing. DNA activity can thus be turned on or off, and tissue differentiation can occur into liver, eye, WBC, muscles, etc. There's also a regulation of transcription in the cytoplasm. Once the mRNA leaves the nucleus and gets into the cytoplasm, there are cytoplasmic proteins that must then enter the nucleus in order to facilitate mRNA and rRNA polymerase for DNA expression. So the transcription factors are those that inhibit mRNA. And the way transcription factors do that is by, again, preventing the mRNA from reading and executing the instructions from DNA in the nucleus. So the transcription factors enter the nucleus and then prevent mRNA, RNA polymerase from doing their jobs. And the cytoplasmic inhibitory complex maintains transcription factors in inactive form in the cytoplasm. And the CICs can be made inactive or active depending on the body's needs, and sometimes can be made rapidly active or inactive as when the cell needs to produce antibodies. This is another term that you'll meet often in modern molecular biology, epigenetic regulation or epigenesis. Epigenesis is from generation to generation. So in that sense, it is hereditary. And epi is from the Greek. It means upon, or it signifies a layer of control above or laid upon the DNA code. The cell is then programmed to function in a particular way. Instructions are then given to the next generation. Epigenetics is therefore the process by which the genetic code is programmed for expression or silencing without changing the basic DNA sequence. Now, the common known ways, and I'm sure there are unknown ways which we haven't discovered yet, but the known ways this is done is by the modification of those histone proteins, I told you about that in a previous slide, or by a process called DNA methylation, where methyl groups are attached to DNA, thus inactivating that part of DNA. So on the top of this diagram or image is methylation of DNA, making that area inactive. In the middle is that familiar coil structure denoting active DNA. And in the bottom of the image is the histone proteins, which have been modified by those green and red things from actually allowing DNA to do its function. Now, we have known, and we have known it for probably more than 100 years, that there are certain things like environmental factors, or we have known epigenesis or epigenetics acting in various situations. For example, some time ago, there was a British study that showed that men who started smoking before puberty increased their risk of fathering overweight boys. There was a Swedish study again several decades ago that showed that Swedish men exposed to famine showed less risk of diabetes and heart disease in offspring. And we know from the advice given to pregnant mothers nowadays that if you have low folate in the mother, that increases the risk of spina bifida and neural defects. How does this happen? Now, we believe that this is because of epigenetics, and rodent models are providing a clue. For example, rats fed a high-fat diet. We can see that in offspring, there's altered methylation of DNA in the pancreas. And then the offspring are also usually obese. So that is an epigenetic trait from environmental factors that is passed on to the progeny. In pre-diabetic mice, there's altered methylation of sperm DNA that increases diabetes risk in the next 2 generations. Cell cycle regulators, you've seen this often, I'm sure, when you're reading about chemotherapy, cancer, cell turnover, all very important. You know that G1 is the gap 1 of the growth phase when the DNA is prepared for replication. Once the cell enters the S phase or the synthetic phase, it's committed to duplication. Once it goes into the G2 phase, we know that it's then going on to the M phase, which is the mitosis and cell division phase. Cyclins are temporarily expressed regulatory proteins that activate another key enzyme, the cyclin-dependent kinases, which are essential for cell division. So cyclin activates cyclin-dependent kinase, and cyclin-dependent kinase sets the cell division process in motion. Now, as you would have realized by now, many different mechanisms exist to control very finely the processes that go on within a cell. CDK is actually one of those systems. For discovering the cyclin-dependent kinase system, Timothy Hunt and Paul Nurse were awarded the Nobel Prize in 2001. And CDK4/6 inhibitors are now approved, as you know, for treatment of metastatic breast cancer. They end with the term cicicilib, bit of a tongue twister. And to round off this section on molecular control of cell function, if you go back to this diagram, you'll see that there's an S phase, or the synthetic phase. Now, during this phase, the cell is committed to DNA duplication, but is preparing for it. And we know that there is another regulatory mechanism with a different set of proteins that can cause a pause in the S phase that allows for DNA repairs before division. It's pretty wonderful. You would now have realized, and I want to spend a couple of moments talking about how molecular biology is making surgery less and less important in the treatment of cancer. And a time is coming, it might even come during your careers, it won't come during my career, but it'll probably come in the latter part of your career, when the treatment of cancer will devolve into writing a prescription in the way that we get a prescription for an antibiotic for a bacterial infection, take it, and the infection is solved. In other words, molecular biology is proceeding at such a rapid pace, that surgery is becoming less and less important in the treatment of cancer. And many would argue that it has already in some ways, given up its central role in the treatment of cancer. And the synthetic molecular antibodies made in labs, the various biological agents, the protein inhibitors, the RNA inhibitors, the DNA inhibitors, all those are becoming more and more important. That's the way it should be. You know, even 20, 30 years from now, perhaps our successors, surgeons, would look back on us and marvel that we actually did things like take out the breast for breast cancer. That would become something of a surgical museum piece. And that's the way it should be. And it's proceeding in that direction. Now, to close this lecture, I want to talk about apoptosis. Apoptosis is programmed and controlled cell death. It's programmed by molecular mechanisms, and it's not cell death from trauma or necrosis, for example, by cutting off the blood supply, ischemia, necrosis. That's a different type of cell death. But if it's programmed in the cell, at the molecular level, it's called apoptosis. Apoptosis is from the Greek, the falling leaves of autumn. And you might know that there are 50 to 70 billion cells that are renewed every day. They die, they are replaced, they die, they're replaced. And in the vast majority of cases, the miracle is that nothing untoward usually happens. Cancer, of course, upsets this mechanism. Apoptosis is programmed to a final common pathway that converges on mitochondria. Apoptosis is mediated, in other words, by mitochondria. The P53 gene, which was actually a very big part of molecular biology research in the early 1990s, until a whole lot of other things became really interesting. Mitochondria lose their membrane integrity. Enzymes called capsases are liberated. The cellular organelles are then degraded within the cell, and thus shielding the cell from inflammatory responses, and then the cell dies. Apoptosis. So that's the end of the series of 2 lectures on molecular biology. I hope I've been able to transmit some of the great excitement of molecular biology to you, and some of the amazing ways in which within a cell there's a whole universe of structures, regulations, similar to the universe itself, of the solar system, the various things acting in coordination and very fine coordination with each other. Again, as the psalmist says, we are fearfully and wonderfully made. The next lecture will be on surgical anatomy. I plan to do a lecture next Thursday on the surgical anatomy of gastrectomy as well as the lesser sac. And then we'll go on to some other topics. Thank you very much.